Hello, everyone. Welcome to today's live broadcast. From small-scale stem cell production to large-scale hiPSC-based drug development, the flexibility of stirred tank bioreactors. I'm Jennifer Mercarian, the manufacturing editor of BioPharm International, and I'll be your moderator for today's event. We are pleased to bring you this webcast presented by BioPharm International and sponsored by Eppendorf and Ncardia. I would like to share a statement from our sponsor. Eppendorf is a leading life science company that develops and sells instruments, consumables, and services for liquid, sample, and cell handling in laboratories worldwide. By exploiting the strong synergies in bioreactor technology and polymer manufacturing, Eppendorf has emerged as a global player and valuable resource to its customers in the bioprocess marketplace. Eppendorf was founded in Hamburg, Germany in 1945 and has more than 3,000 employees worldwide. The company has subsidiaries in 25 countries and is represented in all other markets by distributors. Ncardia is a leading contract research organization in human cardiovascular and neural stem cell drug discovery, encompassing disease modeling, iPSC manufacturing, and iPSC-based screening, operating worldwide with facilities and offices in Belgium, the Netherlands, and the USA. Ncardia is built on the belief and trust that STEM technology will help to get better medicines to patients faster. The goal of the company is to be the supportive and trusted partner for scientists operating in the hiPSC drug discovery and development space, including cardiovascular and neural safety and efficacy studies. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the dock at the bottom of your presentation window. I would now like to introduce today's speakers. We are pleased to be joined today by Ankita Desai, Dr. Arie Reijerkerk, and Dr. Farbod Famili. Ankita Desai is a bioprocess field application specialist at Eppendorf, North America. She supports customers with her expertise in cell culture bioprocessing and application. After earning a master's degree in pharmaceutical sciences from Northeastern University in Boston, she has gained several experiences working for global players in the pharmaceutical industry. Arie Reijerkerk is the head of services and innovation at Ncardia in Leiden, the Netherlands. He holds a master's degree in cell and molecular biology from Wageningen University and a doctorate in the field of angiogenesis and oncology from the University Medical Center Utrecht. Arie has been working for Ncardia since December 2016. Farbod Famili obtained a PhD of immunology from Leiden University Medical Center in the Netherlands. He has strong experience in cell-based assay development, automation, and hit identification in a variety of therapeutic areas. Currently, he is the scientific project leader of drug discovery and high-throughput screening projects at Ncardia. Thank you all for joining us today. Ankita, would you get us started?
Thank you, Jennifer. Hi, everyone. My name is Ankita Desai, and I'm a bioprocess field application specialist with Eppendorf. Today, I am going to be talking about some basic principles involving bioprocess work in my part of the presentation. So some key objectives of this presentation would be to provide some basic introduction as well as key functions of bioreactors, mention some different parameters that would need to be monitored or controlled during culture, as well as how impeller geometry or agitation affects cell growth, as well as a brief overview on perfusion and some other modes of culture, which will be followed by a Q&A session at the end of the presentation. So Eppendorf has a wide variety of product offerings, which includes everything from liquid handling products like pipettes, sample handling products like freezers and centrifuges, among others, as well as cell handling products, which includes bioreactors, which is what we are going to be focusing on today. So stem cell-based technologies are promising approaches for therapy as well as for drug discovery. For commercialization, researchers are constantly evaluating standardization of their cultures as well as efficient scale-up. Eppendorf customers rely on our bioprocess solutions for controlled cultivations of induced pluripotent stem cells or iPSCs, mesenchymal stem cells or MSCs, and others. So stem cells are a subject of great interest in the industry as well as academic research field. Pluripotency as well as regenerative properties of stem cells makes them extremely promising candidates in regenerative medicine as well as drug research. So in this part of the presentation, we are going to talk about how stem cells are produced and used in cell-based therapy and drug discovery, as well as some of the unique challenges that they pose. So the first stem cell application was actually a bone marrow transplant that was performed as early as in 1956. Actually, stem cells can be used to mitigate a wide variety of conditions like wound healing, spinal cord injury, stroke, Alzheimer's disease, just to name a few. So you may have often heard people mention that stem cell expansion as well as scale-up is pretty challenging. And here are some of the reasons why stem cells pose a unique challenge. So first would be spontaneous differentiation. So depending on factors like agitation or shear stress, some culture conditions like pH, oxygen concentration, chemical agents, etc., stem cells can differentiate into different cell types. Another challenge would be aggregation. Since these cells tend to grow as aggregates sometimes, which makes it challenging to culture them as suspension culture. And also, since the final product of such a process would be cells, it is extremely critical that the final stem cell population retain their pluri or multipotency, as well as the ability to differentiate into the desired cell type. This, not to mention, should also produce large number of cells enough to give a meaningful and a therapeutic dose. So all of these factors would then contribute to challenges associated with stem cell expansion as well as scale-up. So routinely, stem cells are cultured at a small scale in 2D formats, which would include plates, tea flasks, etc. However, depending on the therapeutic application, stem cell-based therapies would need around 1E5 to 1E10 cells per treatment per dose per patient, which would again depend on the therapeutic application being targeted. So this requires a large number of cells to be cultured, which in turn increases the number of 2D platforms required. As the amount of tissue culture surface area per lot increases, the enzymatic harvest and downstream processing of hundreds to thousands of liters of cells could lead to new and challenging process bottlenecks. So while next generation adherent current platforms are under development, which specifically target cell therapy applications, some of the available options are bioreactors, hyperdensity stacked vessels, microcarrier-based cultures, or induced suspension adaptation growth methods, especially for primary adherent cell types. So this is where bioreactors are advantageous with respect to scalability, automation, as well as process control. So short tank bioreactors offer various advantages over 2D growth platforms. These would include complete process monitoring and control, giving us the ability to fine-tune and maintain cells at process conditions like oxygen tension, pH, etc., which would help us mimic in vivo conditions within a culture. Bioreactors also provide the ability to scale up to larger volumes with similar vessel geometries at various sizes, providing an ease of manufacturing meaningful therapeutic doses, again depending on the therapeutic application. So bioreactor scale-up is mostly designed based on vessel geometry, including impeller diameter, the vessel diameter, liquid height, and again, the ratios thereof. So these principles, again, can be easily transferred to single-use vessels, which makes it all the more attractive to adapt the scale-up to industrial needs. Bioreactors offer some unique advantages for cells to be grown in culture conditions which mimic physiological conditions. So, for example, bioreactors provide process monitoring and control of various parameters like pH, temperature, dissolved oxygen, etc. So stem cell expansion, particularly in stirred tank bioreactors, is scalable, starting from small scale or R&D systems like the DAS box, which have a maximum working volume of 250 mils, to then larger pilot scale systems for production. So using bioreactors equipped with control software and sensors, various process parameters like pH, dissolved oxygen, temperature can not only be monitored but also controlled at the desired set point. Bioreactors also help control mechanical parameters like agitation and foaming, as well as the flexibility to use different process strategies like batch, fed batch, or perfusion culture, thereby providing many options for researchers. Stirred tank reactors also offer easy scale-up and scale-down approaches due to similarities in vessel geometries, and cells can also be grown in stirred tank reactors on different growth surfaces like coated or uncoated microcarriers or as cell-only aggregates. So here we have mentioned some of the feeding strategies that are available with bioreactor use. So the first among them is batch culture. So in our typical batch culture, initially medium is transferred into the vessel and then inoculated with a culture and left for a period of time. During this culture time, certain critical parameters are monitored as well as controlled during processing, which includes pH, dissolved oxygen, and temperature. So eventually the initial carbon source becomes depleted by culture consumption, and the product is harvested at the end of the culture. Next, we move on to fed batch system, in which system, after the depletion of initial carbon source, an additional feed is added, which could be in the format of pulsating, constant, linearly increasing, or exponential feed. Again, which feed method is to be selected, that would depend on the type of organism as well as the product formation kinetics. In this type of culture, product is recovered during harvest. Next type is a continuous type of culture in which, following the depletion of the initial carbon source, feed is added and harvested at the same flow rate, which results in essentially a steady state operation system. In this particular type of feeding strategy, dilution rate is a critical process parameter, and the product recovery also occurs during the continuous harvest process. And lastly, we have perfusion, which is like the continuous cultivation system. This type of perfusion happens with a cell retention device, in which spent media is constantly removed from the vessel while retaining cells in the system or bioreactor, and then fresh media is constantly added to the vessel. So using this type of a cell retention system is to achieve higher cell densities and avoid stripping cells out of the system. So now we can talk about some of the process parameters that are very critical to bioreactors and cell growth in more detail. So first thing that we'll talk about is going to be pH control. pH is a vital process parameter that affects cell growth as well as production. Physiological pH is in the range of around 7.4, which is also approximately the pH of most commonly used media types. So typically, mammalian cell growth is negatively impacted below a pH of 6.9, creating an acidic environment, as well as above a pH of 7.7, which creates a basic environment. Bioreactors are equipped with pH probes, which are constantly monitoring pH and providing feedback to the controller, which then uses carbon dioxide or base to control pH at the required set point, thereby ensuring that the cells are not exposed to unfavorable pH values. Another important process parameter is a dissolved oxygen control or DO control. Bioreactors are equipped with a dissolved oxygen sensor, which is constantly monitoring and through feedback mechanism controlling dissolved oxygen at a certain set point, which gives optimal cell growth conditions and prevents cells from being exposed to more or less oxygen than required, which could be harmful for the cells. So stem cells can be grown based on an attachment matrix, which is required to be able to grow stem cells in a stirred tank bioreactor. So here we list three different types of attachment matrix. First would be microcarriers, which are essentially spheres of around 100 to 300 microns in size. They can be made of various materials like dextran, polystyrene, glass, or even gelatin. And various materials can be used for surface coating like collagen, which would then enhance the attachment of cells to these microcarriers. The second type of attachment matrix would be fiber cell discs. So these are essentially three-dimensional polyester mesh on which the cells would attach easily and be protected also from shear forces in culture. So this would give an added advantage of the cell being constantly maintained in a low shear environment. And lastly, we have the cell-only aggregates that can be directly cultured in a bioreactor. So the design of the impeller as well as agitation speed are the most effective way to impact shear forces in culture. An optimal agitation speed creates a balance between ensuring a homogeneous suspension and not causing too much shear stress on cells, which could cause stem cells to spontaneously differentiate. The different cell types grown on different surfaces have varying sensitivities. For example, some cells on carriers suffer from more fluid dynamic stress than free cell suspension, probably because they are attached on larger particles that are more prone to collisions, which might damage the cells. Whereas in a macrocarrier suspension, cells are less sensitive to shear forces since they are growing inside carrier beads and may be protected against detrimental shear stress and bead collisions. So agitation needs to be optimized depending on the stem cell type, growth surface, working volume, etc. So here we talk about some of the criteria or parameters that need to be considered during scaling up cell culture. So typically for scale up, tip speed and not agitation is kept constant across the scales. So tip speed influences mixing time as well as oxygen transfer. Thereby, keeping a constant tip speed during scale up helps keep the cells in suspension as well as gives a constant shear stress across scales. However, using a constant tip speed as a scale-up approach may result in reduced mixing time or mixing performance in larger vessels, which is another consideration, something to consider when you actually try to scale up your culture. Impeller geometry is another factor which greatly influences shear stress as well as cell growth. So in order to identify the most suitable impeller geometry for the cultivation of 3D tumor cell aggregates in the DASBox mini bioreactor system, researchers have cultivated 3D spheroids in vessels equipped with a 3-blade impeller with a 30-degree pitch, an 8-blade impeller with a 60-degree pitch, as well as a 6-blade Rushton impeller type. So here we see that different impeller types cause different flow patterns in the reactors and vary in terms of shear stress and oxygen transfer, which in turn would greatly affect cell growth. So pitch blade impeller, like the one we see in the picture here, is typically used for cell culture applications, causes an orthogonal flow. Whereas a Rushton impeller, typically used in microbial applications, causes a horizontal. So process scale-up is typically needed to transfer a process out from R&D scale and to move into pilot production volume. So the goal here is to optimize the process at a small scale and then transfer that to a larger scale in order to get a higher yield with constant product characteristics. So during scale-up, the goal would be to, again, move to a larger scale while you're maintaining constant key process parameters. like shear stress, concentration gradient, oxygen supply, among others. So these key process parameters greatly influence cell physiology, which again would in turn affect product yield as well as quality. So here we mentioned some key factors to keep in mind during process scale-up. The first one is oxygen transfer, which affects the oxygen supply in aerobic culture, which could again be a limiting factor for sufficient cell growth as well as obtaining a good product. Next is mixing time. This is important in order to avoid the concentration gradients throughout culture. Next is tip speed, which affects shear stress, mixing time, as well as oxygen transfer. Next is your power number, which influences, again, oxygen transfer and culture mixing. And more scale-up strategies would aim at keeping one or more parameters that we listed constant across the scales, and that would depend on the process requirements. So the driving force typically for any culture is, typically for cell culture, it would depend on how much of an oxygen concentration gradient is there across the culture. So in order to focus on that, the next thing to consider would be volumetric mass transfer coefficient, or KLA, which is again one of the most commonly looked at parameters while considering scale-up. So this volumetric mass transfer coefficient, or KLA, would be the driving force between the concentration gradient of oxygen at the interface versus that in the bulk liquid. So depending on operating conditions like equipment-specific parameters, namely vessel geometry, stirrer type, installation conditions, or process parameters like agitation rate, filling, aeration rate, type of gassing device, and pressure, or even the properties of the liquid medium like density, viscosity, surface tension, and coalescence properties. So all of these would then in turn affect KLA values. So for proper cell culture scale-up, the idea is to select equipment from different sizes which would offer similar KLA capability. That will offer sufficient overlapping so that small-scale success can be replicated in large scale. So to conclude, in this section of the presentation, we went through a broad range of applications of stem cells in cell-based therapy and drug discovery. We also talked about some unique challenges involving stem cell expansion as well as scale-up, and the ability of bioreactors to constantly monitor and control key process parameters that influence stem cell growth, as well as some benefits of stirred tank bioreactors in providing reproducible conditions and automated process controls with ease of scalability. So that would conclude my part of the presentation. Thank you for your time.
So we'll continue with a case study presented by Ncardia. And first of all, we would like to really thank Eppendorf, our colleagues at Eppendorf, that gave us the opportunity to share what we are doing at Ncardia. And in this presentation, we want to really show how we at Ncardia combine large-scale manufacturing platform of iPSC-derived cells with phenotypic screening for discovery of new drugs. We will particularly focus on cardiac hypertrophy, and results will be presented by Farbod Famili in a later phase during this webinar. The mission of Ncardia is that we believe that human iPSC technology will help to get better medicines to patients faster by improving drug discovery and development process. And what we will do during this webinar is to give you insight into how the advent of human-induced pluripotent stem cell technology has substantially expanded the availability of human cells for drug discovery to uncover the potential for better translation to the clinic and reduction of late-stage drug attrition. At the moment, it's very clear that classical drug discovery and development approaches are expensive and relatively inefficient. And there are multiple reasons underlying that. And what the picture shows here is a well-known funnel that drug developers are facing. Generally, in classic drug discovery programs, large-scale screenings start with simple but not very predictive models, not using human cells generally. Human cells have a limited availability, and another confounding factor which drug developers are facing is batch variation between different batches. Usually the next step, when multiple compounds were not successful in the first screening, the next step is testing in animals. It's increasing costs. It's a gatekeeper for clinical trials, so there's much regulation going on that's dictating animal testing. The experiments are generally complex and time-consuming. Then in the next step, when a drug is still effective in animal models, studies may be initiated in the clinical trials. But it's very clear also from many reviews and publications that poor predictivity in early assays, but also in animal studies, could finally lead to late-stage failures as well, even in clinic. So as said, Ncardia, we believe that iPSC technology can really overcome the issues that we face in classic discovery programs. The beauty of iPSCs is that you generally can generate every cell type of the human body that you can think of in theory. And in Ncardia, we have been particularly investing resources in the setup of large-scale manufacturing and process development of several types of iPSC-derived cell models. For example, pleurocyte cardiomyocytes, but also atrial cardiomyocytes, endothelial cells, smooth muscle cells in the cardiac area, but also many different cell types in the neuronal area. In combination with our disease modeling capabilities and also a great combination of several assay technologies, we provide excellent opportunities for drug discovery. And I'm sure that Farbod will elaborate on this in much more detail when he shows his case study. Consistent, high-quality, large-scale manufacturing of functional iPSC-derived cells is key for implementation of hiPSC-based phenotypic disease models in drug discovery and development. And therefore, central to Ncardia's offerings is a manufacturing platform which is comprised of state-of-the-art bioreactor systems, which allow us for large-scale manufacturing of cells which are required for the applications that we offer to our customers. In this case study, we will not focus too much on the different cell types that we have on the market. We will not focus as well on the services that we can do with those cell types for our customers. We will definitely focus more on Ncardia's industry-leading process development and manufacturing of iPSC-derived cardiomyocytes at large scale. In the second part, Farbod will present a case study using these cells with our hiPSC-based drug screening platform, which we call Discovery, and it will really show exciting results on the generation of disease-relevant human stem cell-derived models, and also development and validation of phenotypic assays, and it will share very exciting screening results. And altogether, while doing this, we really believe that we can bring true human biology to drug discovery and development. Our core manufacturing platform is comprised of state-of-the-art bioreactor systems that allow us to assess and optimize critical process parameters at small scale to validate them at mid-scale and to go for large-scale manufacturing and automated closed systems. During the development of processes for iPSC cardiomyocytes, we have been really focusing on understanding what's going on in those bioreactors. And just as a typical example, I want to show you some intermediate quality attributes that we have been analyzing in this slide. What you clearly can see over the period of differentiation of cardiomyocytes from iPSCs, we assess iPSC markers in early phases, and you can clearly see them fade out. It's very interesting that halfway the differentiation process, already a specific marker of cardiomyocytes called cardiac troponin T is elevated. What is very important to understand is that early levels of troponin T really determine the outcome of the final product quality. And I realize this is just an example of the things that we analyzed during the process. There are many things that we assess in real time, like oxygen requirement, like pH, like the presence of metabolites in medium, which help us to understand the process and which also helps us to generate high quality cells at the very end of the manufacturing process. This slide is just summarizing what we have been achieving so far with our manufacturing platform, showing iPSC cardiomyocytes as an example. So what we have at the moment is a process that allows us for reproducible manufacturing of up to 5 billion cells. What we can do is monitor and control critical process parameters, and that seems to be very key for a robust process. So we assess, as I just showed you in the previous slide, we assess several intermediate critical quality attributes that allows us really to keep the process under control. We implemented automation and a control strategy to ensure high product quality. The manufacturing process that we've been developing so far is very robust and demonstrated reproducibility in the final cell product in different batches. And that's shown in this slide here. What you can see upper left is troponin T levels in multiple batches. We have been producing so far over 20 batches. And troponin T levels are generally very high. The process, as mentioned, is very robust, and we really believe that that's due to the fact that we use controlled bioreactors, that we understand what we're doing in those bioreactors, that we understand what deviations mean in critical quality attributes that we analyze during the process. Very interestingly, we have also used our platform for additional iPSC models, and just below that, just two examples. The process is so robust that we were able to generate high purities of cardiomyocyte batches from different disease lines. And in summary, what we have been achieving so far is that in more than 80% of the batches, troponin T levels were higher than 75%. So from this, I'm very happy to hand over to Farbod Famili, who will really show you a case study about how we use iPSC technologies and our large-scale manufacturing platform for drug discovery.
Thank you, Arie. So the last part of the presentation, I would like to discuss about the launch of our another service, which is actually our drug discovery services. So at Ncardia, using our iPS-derived cellular models, mainly cardiac and neurons, as we showed you, we have two separate services. One is, of course, the manufacturing service that we explained to that. The manufacturing service by itself can also give us the opportunity to, of course, produce the industrial scale cells. That means that give us the opportunity to do also drug discovery services. So since around beginning of 2019, we also officially launched our drug discovery services, which is called Discover It. And in this slide and next slide, I would like to explain to you some data that we obtained during our demonstration study that we did to launch this capability of Ncardia. So the Discover It is actually a full-service drug discovery efficacy screening platform that comprises of 4 modules. The module 1 is disease modeling, and that means that using the iPS-derived cardiac and neuronal cell model, we can use either the patient source or we can use the other sort of mutation induction, like, for instance, CRISPR modification to bring a true disease biology into a screening platform. And then upon generation of this disease model, we can produce the cells at the manufacturing scale, at the industrial scale. We just showed you some data. And the third module would be development of the assay, which is robust and which is reproducible for a large-scale screening using at early drug discovery. And finally, after development of a good assay, we can do compound screening and hit identification and confirmation. So Basically, what Ncardia is offering here is efficacy phenotypic screening over a target-based screening, because we believe that having a phenotypic screening in place by providing a better model which is based on iPS-derived cells and also a more true human biology system can actually reduce the time of early drug discovery, because by that you will have a higher hit rate in your early discovery. That means that you would need actually lower number of compounds to screen. And also the fact that you can do the screening platforms more on really at the cellular level instead of using as actually target-based, that helps to have a more comprehensive picture of your true disease model. And of course, the possibility to develop the patient-specific models, that gives also the opportunity to produce the stratified medicines for your early drug discovery. Therefore, the whole discovery platform can give the opportunity to do a faster drug discovery, increase success rate, but also reduce the cost of the early drug discovery. So here I would like to actually show you some data that we obtained during our demonstration study. For this study, we used a cardiac hypertrophy model to go through the whole discovery platform. So for your information, cardiac hypertrophy model, hypertrophy is actually enlargement of the left ventricular septum. That would be caused due to either hypertension or could be due to a genetic defect. And due to that, the patient might get arrhythmia, and even if it's left untreated, it can lead to death. So this was the model that we used for this case study. We produced actually two different cellular models. One was originated from a healthy donor, but we tried to induce the hypertrophy in a physiologically relevant model that I will explain later to you. And the other line that we generated was a patient-derived line, came from a myosin heavy chain mutated patient that actually was diagnosed by hypertrophy, also due to the hypertension and also arrhythmia. The cardiac cells were generated from these two different models, and they went through the standard procedure that we use normally for characterization of the cells. This is either purity by flow cytometry, for instance, looking at the troponin level, or also the functionality of the models, if applicable, for instance, the electrophysiology or calcium flux assays. In this particular model, we use endothelin-1 as an inducer, as a stimuli. Endothelin-1 is a physiologically relevant basic peptide that is also produced by endothelial cells. And as a readout in this model, we use the BMP. BMP expression is also a biomarker which is very clinically relevant for detection of cardiac hypertrophy in clinic. So this slide actually shows the whole workflow of how we performed the assays and at the end the screening. So the cells were produced as we mentioned at the industrial scale using our manufacturing platform. The cells were QC'd carefully according to the QC procedure that we have, like purity and functionality of the cells. And then the whole actually was performed fully automated using the automated liquid handling systems that we have at Ncardia, allow us to do the full handling of the 384 well plates, like coating, seeding, and medium refreshment, compound addition, all the way up to also data analysis. So what you see here is the timeline of how we handle the cells. So the cells were seeded on 384 well plates. They were refreshed for 6 days, and at day 6, the endothelin-1 as the higher induction of the hypertrophy was added, following by the treatment of the compounds. and the day after the cells were harvested, and two different assays were actually developed. One was the NT-proBNP alpha lyase assay, and the other one was a high content imaging assay that I will explain in the next slide how actually we qualified the assays. So what you see at the left is the high content imaging assay looking at the BMP marker, intracellular BMP expression, and what you see is that upon induction with endothelin-1 there's a significant upregulation of the BMP. And at the right you see the result from the NT-proBNP alpha lyase assay. So also the secretion of the NT-proBNP in the medium was significantly increased. This assay was chosen actually for the screening later on, so from now on I would like to focus on this assay. So what you see here is that upon induction of the endothelin-1 there is an assay window of 17 to upregulate the secretion of NT-proBNP. Verapamil was used here as a positive control, so by the treatment of the cells on the verapamil, what you see here is that the phenotype is reversed and the secretion of the NT-proBNP is come back to a basal level. Furthermore, we did extensive basically reproducibility and robustness analysis on the assay to optimize it. We looked at the QC criteria like assay window signal to background ratio, coefficient variation of the controls, and also the Z factor. So at the end, the outcome was that the developed assay had a signal to background ratio of about 4. What you see here exactly is 17. The very good reproducibility, so the percent CV of below 20%, and also the Z factor of above 0.4. Next, we did the assay validation. So we screened a selective panel of compounds, around 90 compounds that they were screened in duplicate. We chose both poro and anti-hypertrophic compounds at 3 different concentrations, 0.1, 1, and 10 micromolar. And the idea was that to validate the assay in the final screening format and see if we can still repeat the QC criteria that we obtained in the previous slide, but also to see that how reproducible are the compound effects. And it turned out that actually the developed assay is very robust, because the Pearson correlation of the duplicate plates in all concentrations showed the Pearson of above 0.9. And also we could repeat basically all the QC criteria that we obtained during assay development. So we were very confident that the developed assay is ready to go to the screening. So here I would like to first show you actually a summary of what we did during our high throughput campaign and what we obtained, and later on in the next few slides I will more elaborate on that. So we performed the full basically drug discovery phases, primary screen, heat confirmation, and at the end the potency analysis. In total we screened around 10,000 data points from the libraries that were basically focused, the libraries that have high heat rate and very high diversity. So in total around 4,000 compounds were screened at concentration of 1 micromolar, and we set a cutoff of around 40% at the primary screen to perform the heat identification. From that we obtained around 300 heats that were confirmed via different assays during the heat confirmation phase. We confirmed first the heats to repeat in the alpha lyase assay in another batch that resulted in the confirmation of around 248 compounds, and later on we identified the false positives and also in an orthogonal assay, which was our high content imaging assay. And finally, with 192 confirmed heats, which is around like 5% heat rate, we performed the potency analysis, which means we looked at the IC50 curves between two lines, so the healthy line and the disease line that I mentioned. So let's look at this data in more depth. So what you see here is the summary of our primary screen. So the first primary screen was performing 15 screening plates to screen around 4,000 compounds. What you see in the left is the QC criteria of the plates. All 15 plates actually passed the QC criteria in the first round, so there was no need to repeat the screening. What you see here is the percent CV Z factor of the plates and also the assay window. So the screening resulted in the signal to background of around 15 and Z factor above 0.4. And what you see at the right is the distribution plot of the all compounds. So each dot is basically one compound which is normalized to the positive control. The red dots are positive control, so they are set to 100% inhibition. And based on the distribution analysis that we did during the validation screening, we set the 40% as a cutoff. So all the hits that had above 40% inhibition were considered as a hit. So next we try to confirm basically the initial hits. As I mentioned, 341 hits were identified. We confirmed the hits via 3 different ways. First, we confirmed them in a different batch, again using the same assay, antiprogram alpha lysa assay. From that, we got 248 compounds confirmed, and then we try to eliminate the false positives, so the compounds that are interfering basically with alpha technology. And then from that, we also ended up with 192 final hits. We also performed the orthogonal screening, which was a high-content imaging. That's what you see at the far right. And we saw also some compounds that actually have effect on both assays, so the alpha lysa and high-content imaging, and some that only in one assay, but we didn't take into account the result of this assay for really the final hit confirmation. So eventually we did the potency analysis with the confirmed hits, so 192 hits. We did the concentration response curves of 1 nanomolar to 5 micromolar, and what you see here is that the compounds with different potency were identified. Some had really at the sub-nanomolar potency and lead properties, meaning that efficacy and safety or pharmacokinetic and dynamics are tested already, because most of them were the compounds that were already tested in clinical trials. So basically the screening at the end results on the leads that were already confirmed. And also what you see here is that there is a very good reproducibility between 2 lines. So the top row is the healthy line and the bottom row is the diseased line, and both show the very similar IC50 curve. And by that, I would like to conclude so I hope that you were convinced that at Ncardia we are capable of developing really the manufacturing discovery services and also drug discovery services. So we have a large-scale iPS-based manufacturing platform. That was the first part of the presentation that Arie explained to you. And we also use actually the industrial scale cells to use in a discovery platform, which is our drug discovery services, by developing disease-relevant models using the iPS technology at Ncardia, and developing the predictive assays, which are also robust for screening. And finally, we showed you our demonstration study to do a really hit confirmation and potency analysis using the model of the cardiac hypertrophy, which at the end resulted in identification of compounds with lead properties. So that is basically the capability of the Ncardia as a CMO and CRO expert. So by that, I would like to end this presentation. I would like to thank you for your attention, and we are happy to get any question if there is. Thanks.
Thank you all for such informative presentations. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of the presentation window. Okay, and our first question, Ankita, why are cells grown on microcarriers more stressed by shear forces compared to the cells on fiber cell discs?
Well, some cells on microcarriers suffer from a more fluid dynamic stress as compared to the free cell suspension, probably because they are attached on larger particles, which are more prone to collisions, which then damage the cell. However, when cells are grown on fibrocell disc matrix, they are shielded from the turbulence caused by the impeller rotation as well as the spark.
Okay, thank you. A question for Ncardia. How can Ncardia's platform be used for other iPSC-derived cell types?
Yeah, thank you very much for this question. Thank you also for joining this webinar. My name is Arie Reijerkerk, and I'm happy to discuss it with you. I hope it was clear from the presentation that we have developed processes for iPSC cardiomyocytes. What you also may have seen from one of the slides is that we not only use this cardiomyocytes for internal work for services that we do, but we also offer our manufacturing platform for process development, and in particular process development for iPSC-derived cells. Over the last years, we have gained a lot of expertise about what is critical for iPSC-derived cell production. The reality actually today is that we already applied our knowledge for the generation of iPSC-derived endothelial cells, which is another cardiovascular cell type. So there's definitely multiple options for that we can work together with customers to generate different iPSC cell types. Usually, how we would approach this is that we discuss with the customer what the targets are. We figure out whether there are protocols available that can be transferred to our company. We dedicate a very specific team to carry out the work, to formulate a proposal, to work on timelines, go, no-go decisions. And then again, we leverage our expertise that we've obtained so far with large-scale manufacturing for iPSC cardiomyocytes. So summarizing, there are multiple opportunities here. We have gained so much expertise that we're sure we can leverage for other cell types. And we have one example already in-house, which is iPSC-derived endothelial cells.
Okay, thank you. So another question for Ncardia. What are the opportunities for using iPSC-derived neuron discovery projects, and when do you expect that to be a commonly accessible option?
Yes, thank you very much for the question. So as far as I know, indeed, the literature, there are different reports using the iPS-derived models to develop neuronal diseases. For example, the models like Alzheimer, Parkinson, or ALS. These models are either derived from patients or they are developed by genetic modification, like for instance with CRISPR. As you know, also at Ncardia, we have a cellular portfolio including different type of neuronal cells. For example, we have the type of cells that's called CNS for you, and that's the GABAergic and glutaminergic neuronal cells. But also we have, for instance, cortical neuron cells, astrocytes, all iPS-derived. So basically, in terms of the cellular resource, we have at Ncardia the capability to manufacture these kind of cells and use it for the screening. Currently, we are also ourselves in-house trying to develop also screening platforms for different neuronal diseases that can actually address neurodegeneration and inflammation. But in principle, based on the cellular resource that we have in our portfolio and the capability that we just show in the presentation, I mean, the capability on the screening and the different readouts that we have in-house, we are ready to actually start any collaboration when needed using our cell sources. But as I said again, we are also trying to, as soon as possible, develop our own screening platform on a specific neurodegenerative disease as well.
Okay, thank you. I think this is a question for Ankita. This person says, I didn't understand the KLA value. Is this vessel specific or does it depend on the experimental setup?
Sure, thanks for the question. So KLA values will depend on different parameters. So specifically, it would depend on equipment-specific parameters like vessel geometry, stirrer type, installation conditions, or process parameters like the agitation rate, aeration type, aeration rate, or type of gassing device and the pressure, as well as properties of the liquid medium, like the density or viscosity, as well as surface tension and coalescence property.
Okay, thank you. And then another question about Eppendorf bioreactors. Are there possibilities for online analytics process parameters in addition to the standard parameters?
Sure, yes, definitely. So the DASWare software itself allows for the integration of third-party analyzers. So, for example, Raman analyzers, autosamplers, biomass sensors, and many more.
Okay. So let's see. What are the challenges that you're seeing in the field of incorporating hiPSC-derived cells in screening? Is access to cell lines and reproducibility and manufacturing differentiated cells still a hurdle?
So thanks again for the question. Sorry. I would say indeed the access to the cell lines, especially if it's going to be a patient-derived cell line, is still, since of course we would like to also commercialize them, then you have of course the issues of informed consent, etc. That would take a while and of course still a challenge. And the other thing is that of course different lines, when you optimize a differentiation protocol in your manufacturing system for one line, that should not necessarily work for other lines. So of course you still need some optimization for the differentiation and manufacturing process. But from our experience, when we were manufacturing two different lines of what you saw in the presentation, the control line and the myosin heavy chain line, later on after the optimization, we saw actually very good reproducibility for both lines, resulted into also very similar data, the screening data that we obtained from these two lines. So I would say, yeah, so still you need some optimization, and the access to the cell line is still not easy, but with our large scale manufacturing system and the robust screening platform, you can expect a great result.
Okay. And another question about Ncardia's process. Is Ncardia's process for the commercial hiPSC-derived cardiomyocytes suitable for generation of CMs from other sources as well?
Yeah, this is Arie again, and this is really an interesting question, a very good question. So I assume that this question comes from someone that has, for example, patient iPSCs and that wants to generate cardiomyocytes out of those cells. And that's exactly also how we position our platform and manufacturing capabilities. So what I already quickly touched upon in one of the slides was that we used the process that we have been generating for iPSC cardiomyocytes to generate cardiomyocytes from other iPSC lines. And I think I brought up two examples, myosin heavy chain and also JNLS. And so far, the process has been very successful also for other iPSC types. What you've seen is two examples. At the moment, we are over 10. In particular, we do these manufacturings for customers that want to produce cardiomyocytes from disease iPSCs, either obtained from patients or generated by genetic modification. So there's definitely lots of opportunities there. The process is robust for one line that we use ourselves at Ncardia, and it has been successful for different iPSC lines, over 10. To my knowledge today, 2 out of 12 lines failed, and finally we figured out there were good reasons and we had a good understanding about why some iPSCs don't work and others do. But summarizing, the majority are successful in our process. Thanks for the question. It's very interesting.
Okay. Very good. Well, I think we have time for just one more question. What is meant with scale in and scale out strategy?
So a scale in or scale out, these are different strategies that you can use for scaling up or scaling down your bioreactor size. So by a scale out strategy, it means to be able to use multiple bioreactors at a production scale, which are of the same size as the bioreactors that you would use in the early stage clinical trial. And scale in would just mean the vice versa. The main advantage of this strategy is that you would have little risk for process as well as product quality as you move to different scales.
Okay. Well, unfortunately, we're out of time, but I want to thank the audience for attending and for participating in today's event and for the questions. And I want to thank our sponsors, Eppendorf and Ncardia, for making today's educational webcast possible. So you will receive an email alerting you when this webcast will be available for replay, and we invite you to forward that announcement to your colleagues who may have missed today's live event. Thanks to all for joining, and we will see you next time. Goodbye.